So I'm Dr. Nicola Kingswell. I'm here today to talk to you about how we've been working with Atelerix WellReady to help ship our Labskin 3D human tissue models all around the world. So what is Labskin? As a company, we are specialists in skin science and microbiology, and our model is a 3D human skin equivalent that we grow in the labs. It's a full thickness model that fully supports the skin microbiome. The technology originated from Leeds University, was developed by microbiologists with the sole purpose of studying the skin microbiome. They wanted a better way of looking at the skin microbiome other than agar plates and shake flasks. The technology spun out into the company that we are now. And we use it for all sorts of things, not just the microbiome. So we look at things like skincare, cosmetics, personal care products, anything that you put on the skin and that will interact with the skin, we can look at and study the effects on the skin. It's been developed over 16 years with more than 30, probably nearer 40 now, scientific journal publications. And we work with a lot of industry-leading skincare, cosmetics, personal care, wound healing and therapeutic brands to support new product development, claims and screening. The model is constructed from primary human skin fibroblasts and keratinocytes, and it's living, it's metabolically active with dermal and epidermal components. We use a fibrin-based dermal gel to create the dermal layer into which we seed fibroblast cells, and they're active. So they produce their own extracellular matrix, their own collagen. And then we get a fully differentiated, multi-layered and stratified epidermis and stratum corneum. So that's the dry, flaky top layer that we get on our skin. We can produce large numbers of models, so they're all uniform, high quality, highly reproducible, and it is reactive. So if the keratinocytes encounter something they don't like, they produce cytokines, so we can measure those irritation, inflammation responses in the skin. It does have a dry top surface, and that allows us to put products, including the skin microbiome, directly onto the surface. And it's a suitable model for transepidermal penetration, skin localization studies. And as previously mentioned, we can colonize it with commensal and pathogenic microorganisms for studying skin microbe interactions. So I could even swab your cheek and put your whole microbiome onto the Labskin, and it wouldn't just sit there, it actually grows, it interacts, it behaves just like a microbiome on human skin. And the model expresses key markers, things like Ki67, filaggrin, keratin and kallikreins. And if we take a slice through Labskin, this is what it looks like. So this is a cross-section through Labskin here. We've got a nice dermal layer at the bottom with the fibroblasts busy producing the extracellular matrix. Then we get the fully differentiated stratum corneum at the top. And this is how Labskin compares to human skin. So on the left panel here, we have a human skin explant, and here we have Labskin. So you can see the morphology of the structure is very similar. And similarly, if we look for the expression of key proteins, human skin on the top, Labskin on the bottom, so things like cytokeratin, filaggrin, and laminin and procollagen, which are important skin bioproteins, they're all expressed. Importantly for our model, we produce antimicrobial peptides. So beta defensin and RNA7 are expressed in the model, and this is what allows us to put microbes onto the surface of Labskin. So when Labskin technology first spun out into being a business, that was a long time ago, the whole model, the whole business model was to produce and ship plates of Labskin to researchers that wanted to use it. And that's great, but you're shipping a live product. So as I've mentioned previously, it's metabolically active, it is alive. We have a 2 to 3 week testing window once it reaches maturity. And it needs to be fed, just like we do. It needs its daily media, its daily protein shake. So shipping internationally can be quite tricky. And the way we got around this in the past is we'd create a gel using agar and DMEM media. So there's some nutrients in there, but it would hold the transwell inserts kind of fast in the cells, in the wells, and just allow the model to be shipped. But you got a maximum of 96 hours before we start to see tissue degradation. It absolutely needs feeding at that point. So custom delays, flight delays, bad weather, That results in non-viable Labskin, and that equals sad customers, and that results in us having to send out more Labskin, and it just perpetuates a real cycle of unhappiness, so we say. So the solution was Atelerix WellReady, and they came to our rescue. So we embed the bottom of our Transwell insert into the WellReady gel. We apply a thin layer of WellReady gel to the top of the Labskin, and we supply the customers with a vial of the dissolution solution with the kit, and we apply them with full instructions as well on what to do, because the last thing we want them to do is to puncture the Labskin with a pipette tip and flood it with dissolution solution. And then we ship them, and that's how they ship. We can ship them at ambient temperature, and we've been using the WellReady to do some shipments all around the world. In fact, it came to the rescue a couple of weeks ago, if you remember, that the east coast of the US was hit by really bad snowstorms. So we were shipping to the US, and Newark Airport was closed due to really heavy snowfall. So our shipment of Labskin was stranded there for an extra day. And once it reached the customer, it was absolutely fine because it had been sent with Atelerix WellReady. So this is just a comparison that we've done with looking at Labskin shipped over a number of days. And we mimicked this in the lab by just storing it in a cupboard, a temperature controlled cupboard. So it's either Labskin embedded in Atelerix at the top, or the control. This is just with the DMEM with the aggregate in there. And then we looked at various time points, so 24 hours shipping, 48, 72, 96, and then 120. And we've actually taken this out a little bit further now as well to prove that it maintains it for much longer. And as you can see, we're starting to show degradation here with the 96 and the 120. The structure's all gone horribly wrong. But with the Atelerix, it's very nicely maintained throughout. And again, this is just looking at the Atelerix and the agarose just to see how far we could push it out. And we compare this to a QA sample. So this is one that we take on day 12. This is what we class as our time point of maturity. And just to show that we can use Atelerix to ship that bit longer and maintain the structure of the Labskin. Obviously, we use the skin for microbiome studies, so it was important to us that the integrity was maintained with the ability to populate the surface with microbiome. So we looked at three key components of the skin microbiome, so C. acnes, C. striatum, and S. epidermidis. These three microbes make up about 80% of everybody's skin microbiome. They just appear in different ratios depending on who you are. So we applied them to the surface of the Labskin, to Labskin that was with Atelerix, with the agarose DMEM, or with standard DMEM maintenance media. And then we looked at the enumeration of the microbes. So was the skin integrity maintained to allow these microbes to populate on the surface? And you can see that, yeah, really well. The Atelerix mimicked the model. In fact, probably a little bit better. It supported that growth really well. So we have no qualms in Labskin that's been treated with Atelerix to be able to continue to do those microbiome studies. So I hope you found that interesting. We will continue to ship with WellReady. We have lots of planned shipments coming up all around the globe. For us as a company, it's just given us that peace of mind that we can ship anywhere around the world, that we don't have to worry about those custom delays, that we don't have to worry about those acts of God that may delay our shipments to our clients. Feedback from our clients has been very positive as well. They've all found that the dissolution solution's easy to use. We've walked them through that they may need to leave it on a little bit longer than initially specified, just to make sure that it has all been removed from the surface of the Labskin. If you have any questions about the Labskin or our use with the Atelerix WellReady, please do get in touch.